Hello and welcome to this series of lectures in Essentials of Immunology. To summarize the previous lecture, we had looked at how to establish homozygosity for the breeding of inbred strains of mice, the generation of congenic resistant mouse strains that differ only at the MHC locus, and also the generation of recombinant mouse strains that differed within the MHC locus, which actually led to the discovery of the two types of MHC, MHC1 and MHC2, and how they determined the proliferative versus cytotoxic T cell assays. Now, going into the structure of the MHC antigens, we also learned how the MHC1 antigen or the MHC1 molecule has got a heavy chain of 45 kilodaltons, which is associated with a lighter subunit of 12 kilodaltons called as the beta 2 microglobulin, and how only the heavy chain was anchored into the membrane and the different domains of this heavy chain alpha 1, alpha 2 and alpha 3. In contrast, the MHC2 molecule had two subunits, both of which were anchored into the membrane. These two subunits being the alpha 1, which was 33 kilodaltons in size, and the beta subunit, which was 28 kilodaltons in size. The alpha subunits having two domains, alpha 1 and alpha 2, and the beta subunits having two domains, beta 1 and beta 2. And we also took note of the fact that the structure was represented this way, having two alpha helices and a floor in both the cases, and the floor having a cleft which we refer to as the peptide binding cleft, which would bind the peptide antigen. So, looking at the variability plots of these subunits, both for the MHC1 as well as for the MHC2, and in this case especially the MHC1 variability, we saw how the alpha 1 and alpha 2 domains were more variable in nature compared to the alpha 3 domain, which was just above the membrane. If one looks at the MHC molecule from the top, that is, if you look at this structure from the top, this is the view that you will get. This is the model that has been arrived at, looking at the X-ray diffractions patterns of all of the HLA molecules, especially in the humans. The HLA molecule was purified and then the detailed structure was worked out, and this is the reason why the structure is written with two alpha helices, because there are two alpha helices over here, and then the beta pleated floor is made up by 7 beta pleats, as you see in the numbers in the blue arrows here, 1, 2, 3, 4, 5, 6 and 7, and these are anti-parallel in nature. And if you looked at the variability of the amino acid residues, they all conform to these circles that are found here on the alpha helix, and you also find the variable residues in the beta pleated floor, the blue circles that has been represented over here. These are the two disulfide bonds that you see over here. This is the N terminal portion and this is the C terminal portion. Of course, the N terminal portion, the C terminal portion continues into the third alpha 3 domain and then gets into the membrane. Since this is the top view, you are not looking, you will not be able to look at the alpha 3 domain. So, this shows us how the sides of this molecule are variable as well as the floor, and this yellow background that has been represented over here is the place where the antigenic peptide sits within the groove of the MHC. This groove is called as the antigen binding groove or the peptide binding cleft. Now, the side chains of the amino acids are in contact with this beta pleated floor as well as the alpha helix. So, one can imagine that the variable nature of the amino acids in these two alpha helices and the beta pleated floor will also vary with the different alleles of the MHC, the haplotype of the inbred strain of mouse, or for that matter the allele of the HLA complex, you will find that these amino acids are different and therefore, would accommodate a different the peptide that is accommodated within this groove will have a different property to it. And this then leads to the diversity in the nature of the peptides that are binding to the MHC antigen binding groove. This variability is what enhances the diversity of the recognition by the immune system. So, going on further to look at this molecule in a different way as to ask the question, does the MHC molecule bind the antigen or its fragments, because we have alluded to the groove as the antigen binding groove or the peptide binding cleft. Especially the observation that immune responses are MHC restricted, and that is the recognition along with the self MHC allele takes place, and that the processing or proteolysis of the antigen was needed before the activation of T cell hybridomas or T cells that would then lead to their proliferation. This was referred to in lecture number 3 as to what these macrophages do to the antigens that they encounter. Because of these two observations, they hypothesized that the antigen presenting cell was taking up the antigen and fragmenting this antigen by proteolysis within the lysosomes or the endosomes. Then this proteolized fragment was then brought out on the cell surface somehow in association with the MHC complex. So, to look at this, various experiments were done to demonstrate the binding of the MHC molecule with the antigen or the proteolytic fragment derived from the antigen that the immune system was exposed to or the macrophage was exposed to. And this one of these experiments was the equilibrium dialysis experiment, and you all know that a simple dialysis experiment involves the diffusion of the low molecular weight substances from inside the dialysis bag to the outside. In fact, this is a method in protein enzymology and when during the purification of proteins, where you want to exchange the buffers that were used with a different buffer, one dialyzes a solution containing a high salt buffer with a low salt buffer outside the dialysis bag, and these low molecular weight buffer salts move out from the bag into the outside, and therefore, you establish a equilibrium, and the higher protein molecules do not cross the membrane, because this membrane is semi permeable in nature, and you have different dialysis bags of different cutoffs like 8000, 12,000 and so on and so forth. 8000 meaning it would retain protein molecules that are below 8000 that are above 8000 and allow the molecules that are relatively below 8000 to move back and forth. So, in this example we have starting from here, the central figure, you have about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Just as an example to demonstrate to you, there are 12 small molecules, and if you allow them to diffuse through the membrane, you have 6 of them coming out, 1, 2, 3, 4, 5, 6, and at equilibrium there would be 6 molecules outside and 6 inside the bag, in addition to the protein molecules of higher molecular size which will not move out of the bag. Now, instead of doing a simple dialysis, this equilibrium dialysis experiment that was used to demonstrate the binding of antigenic peptide to the MHC utilized this principle, where if you consider that these protein molecules that are inside the dialysis bag given here as squares, if these were to be assumed as receptors and you were dialyzing out the specific ligand that was binding to this receptor, one would find that in these 12 molecules putatively being the ligand for this receptor, if there are 4 protein molecules or 4 receptors, it would bind these 4 ligands, and therefore, out of the 12 you would have only the remaining one of them being available for dialysis. So, in this particular case you have 10 ligand molecules, and out of the 10, 4 are bound by this receptor, and therefore, there were only 6 that would be available for diffusion or exchange, and they would equilibrate 3 inside the bag and 3 outside. And with further changes of the outside solution, even these 3 would come out. But essentially, to look at this, you find that if you had a radioactive ligand outside, you put it outside into the beaker outside of the dialysis bag, and if these were to be radioactively labeled, you would find that these molecules would progressively move inside and accumulate in the bag, because they would be bound by the receptor and therefore become unavailable for dialysis. So, basically then the principle would be to look at having purified MHC molecules inside the dialysis bag and put the radioactively or fluorophore coupled ligand or antigenic fragment outside of the dialysis bag, and look at the movement of the radioactivity or the fluorophore into the dialysis bag, and then plot versus time, because this is called as equilibrium dialysis. You allow it to go to equilibrium and look at the number of counts or the fluorescence that has accumulated within the bag that has the purified MHC molecule. This was essentially the experiment that was done to demonstrate the binding of ligand by the MHC molecule. In addition to these, we need to take into consideration certain other observations that allowed this experiment to be done, and that was, as I referred to in the end of the previous lecture, that peptide immunogenicity and antigenicity correlated in certain specific inbred strains of mice. Now, immunogenicity is the term that is used to characterize those substances that were good immunogens. That would mean that the injection of this substance into animals would lead to a good immune response in terms of both T cells as well as antibodies. Now, the term antigenicity, peptide immunogenicity and antigenic immunogenicity, supposing you were to use an antigen that was smaller fragments of a particular peptide, and this antigenicity would mean that it would also bind to the antibodies that were made, and this there is a small mistake here in this slide. This is not antigenic immunogenicity, this should read as peptide immunogenicity and antigenicity. So, these peptide immunogenicity and antigenicity correlated in specific strains. The experiments that were done with certain model antigens like hen egg lysozyme, as I told you earlier, one would immunize hen egg lysozyme, the whole molecule, into a particular inbred strain of mouse, and then go on preparing smaller and smaller fragments of this hen egg lysozyme to see whether these smaller fragments were also immunogenic, and they found that the hen egg lysozyme fragment 46 to 61 turned out to be both immunogenic and antigenic in H2K strains of mice, but not in H2D strains of mice. In other words, H2D would not respond very well to this particular fragment, but the H2K strain would respond very well to this fragment by making antibodies and also activating T cells. So, this 46 to 61 had the ability to bind to T cells and activate them after being presented. Activation of T cells, as we learnt in lecture number 3, would lead to the proliferation of T cells. So, the preparation of T cell hybridomas involved the preparation of immortal T cell clones that had been immunized with 46 to 61 from the H2K strain of mouse. Now, these T cell hybridoma retained their ability to recognize HEL 46 to 61 and respond by the secretion of IL-2, which was one of the consequences of recognition by the structure that recognizes the presented HEL 46 to 61 on self antigen presenting cells, or HEL 46 to 61 that was associated with the H2K molecule on the surface of antigen presenting cells. So, once they had these reagents, they coupled the amino terminus of HEL 46 to 61 with a fluorophore called as NBDF. We will not go into the nature of this fluorophore, except to remember that it is fluorescent. So, this is the HEL 46 to 61, the amino acids that are present in hen egg lysozyme 46 to 61. They also prepared HEL 46 to 61, which had certain replacements in 1 or 2 positions that led to the abrogation of it being a good antigen in H2K mice. So, what was the result that was obtained when they used these reagents? So, again on the top you will find HEL 46 to 61, and they used 46 to 61 with the mutated amino acids as an analog. Now, looking at the T cell proliferation or the release of IL-2, which was again measured by using an IL-2 specific cell line, which would proliferate in response to any presence of IL-2. This is called as a bioassay. So, looking at the thymidine incorporation of the IL-2 specific cell line would give one an idea as to how much IL-2 would be liberated. So, as you had more and more of the antigen on the x axis, and the CPM indicates the proliferation of the IL-2 specific cell. So, when these T cells were exposed to in H2K mice, H2K T cell hybridomas were exposed to HEL 46 to 61, you will see that this is the nature of the IL-2 secretion, which is measured by CPM incorporation by a cell line that is responsive to the liberated IL-2. When the HEL or the hen egg lysozyme is modified by the fluorophore, you would find the same result in that it was very efficient in stimulating IL-2 release. Again, to reiterate, IL-2 release and CPM incorporation are similar, or they go hand in hand in this particular assay. So, once having made sure that the modified HEL 46 to 61 referring to the modification that was done by coupling the fluorophore was as efficient as the native HEL in stimulating the IL-2 release from a T cell hybridoma that was specifically recognizing HEL 46 to 61 in association with the H2K molecule. So, then they went on to do the equilibrium dialysis experiment using large amounts of purified class 2 molecule, especially the IA of K molecule. That means, the IA molecule was expressed using recombinant DNA methods. This recombinant IA molecule that was having the K haplotype or having the amino acid sequence that is present in the K haplotype mice was used to look at how it bound to HEL 46 to 61 by using the fluorophore coupled HEL 46 to 61. So, basically you are looking at the amount of fluorescence that is going in from outside into the inside of the bag. So, let us say you have IA molecule that is purified inside, and you have the fluorophore coupled HEL 46 to 61, which I will put as a star. So, you are looking at this star going into the bag. This star then gets coupled to the IA of K molecule, it binds the HEL 46 to 61. So, therefore, you will have more fluorescence inside the dialysis bag as time goes on. So, at equilibrium you would have to have more fluorescence inside the bag than outside, but at the beginning of the experiment you would have more stars outside the experiment, because you had introduced the fluorescent HCl molecule 46 to 61 outside of the dialysis bag. So, looking at this, it was observed that when you had IA of k inside the bag and the fluorescence went into the bag. So, you had more of fluorescence over here as indicated by these numbers over here, and when you used along with the fluorescent HCl 46 to 61, you mix them with non-fluorescent or native 46 to 61, which would not be having the fluorophore and therefore, not having the fluorescence molecule. It competed for the labeled HCl 46 to 61. In other words, if you had the star marks without any stars, mixed up without any stars, there would be competition between the two, and therefore, you had a competition of the fluorescence from the high fluorescence to the low fluorescence. On the other hand, if they used an analog which had been mutated at the residues which would not allow it to bind to the class 2 molecule, then also you had the star or the fluorescent HCl 46 to 61 going inside and binding to the IA of k molecule. On the other hand, if you use the one that it was not the strain of mouse or the purified IA of molecule that was derived from the strain of mouse that would not respond to HCl 46 to 61, you found that there was no accumulation within the bag at all. In other words, this experiment actually demonstrated that the IA of k molecule would bind to HCl 46 to 61. So, these were some of the experiments that were done in order to look at or demonstrate the binding by purified class 2 molecules, that they actually would bind to the ligand that they were specific to. Now, looking at the structure of MHC1, in contrast to the MHC2 molecule that was used in the equilibrium dialysis experiments, the MHC1 could not be demonstrated as being successful in the equilibrium dialysis experiments. This had to be arrived at by looking at the X-ray diffraction patterns of the crystallized MHC1 molecule, especially the HLA molecule, where they found certain patterns that were characteristic of a peptide sitting in that groove. Now, why is it that the MHC1 did not succeed in equilibrium dialysis experiments? The reason being, the difference between the MHC1 and the MHC2 molecule is in the nature of how these alpha helices are arranged. In the MHC2 molecule, these two alpha helices are said to be open. In other words, the distance between these two ends are little farther apart, and as a result you would be able to look at a peptide that would sit across this groove and extend across the alpha helices. Whereas, in the case of MHC1, the peptide, since the groove was closed at the ends, the peptide had to be located within this groove and could not extend outside. And therefore, you will see that if you look at this MHC class 1 molecule, you see the presence of certain grooves, which is there even in class 2 molecule. These grooves, these pockets are called as A, B, C, D, E and F. So, there are 6 pockets over here, and these are the beta pleated fluor, as I mentioned to you earlier. These are the beta sheets, anti-parallel in nature. So, if you look at these pockets are actually binding the peptides that are sitting within this groove in the class 1 molecule. For example, the A pocket binds to the first peptide residue, the B pocket binds to the second residue, and the C pocket actually binds to the 6th residue. Note the difference, the C pocket is binding to the 6th residue, whereas the D1 binds to the P3 residue. and so on and so forth. So, these are actually called as anchor residues in different alleles of the MHC molecule. In other words, the peptide for the class 1 molecule has to first anchor into these pockets, and when they anchor into these pockets, depending upon the nature of the side chains, you will have a bulge. So, I have demonstrated this the groove here as in the previous slide as a yellow background over here. So, you are looking at this peptide sitting from the top, and when you look at it from the side, the peptide actually shows the 4 and 5 coming outside or raising above this particular cleft. So, in other words, these 3 residues are in contact with the floor, and these 4 and 5 are not in contact with the floor. So, if you look at the MHC class 1 again, as I told you earlier, these are from the side. In order to visualize the alpha 3 domain, which you cannot visualize when you look at it from the top, you see the alpha 3 domain in the blue over here. This is the beta 2 microglobulin that is helping the class 1 structure stabilize on the surface, and this is the place where the peptide is sitting. And to make the future classes a little easier in concept, the T cell receptor, which is supposed to recognize this peptide that is associated with the class 1 or the class 2 molecule, comes and recognizes in this fashion. This is the T cell receptor, and therefore, the T cell receptor, this is the T cell receptor, and this is the antigenic peptide. Therefore, the T cell receptor has to recognize the class 1 molecule as well as the class 1 molecule as well as the antigenic peptide fragment that is derived by proteolysis by the antigen presenting cell. This is the reason why MHC restriction occurs. In other words, this antigen that has to be recognized only in association with this particular allele of the MHC. So, coming back to the variability of amino acids, if this particular alpha helices are variable, that means they would have to bind to different peptides, and different peptides are recognized by different variable regions of the T cell receptor molecule. The T cell receptor molecule is somewhat similar to the immunoglobulin molecule in that they have variable regions and constant regions. So, this then ensures further diversity in the immune system, in the sense that diversity is associated not only with the variable nature of the T cell receptor molecule, but there is further diversity in terms of different antigenic peptide being bound by the MHC allele. So, if you had a peptide of a low affinity to this MHC allele, the T cell receptor would not recognize it with the same high affinity as it would recognize a peptide molecule that is bound with high affinity by the MHC specific MHC class 1 or the class 2 allele. So, this is the nature of how the antigenic fragment is recognized by the self MHC molecule, and this complex leads to the recognition by the T cell receptor. So, when they looked at the nature of antigenic fragments that was bound by the MHC molecule, they found that in the class 1 molecule, because of the closed nature of the groove, one had to elute these antigenic peptides sitting within this groove of MHC class 1 molecule by using acid. This acid eluted peptide fragments were then sequenced by mass spectrometry, by purified by HPLC, and then sequenced by mass spectrophotometric methods, and they found certain very unique properties to the peptides that are associated by different MHC alleles or different MHC proteins, especially the class 1 molecule. They found that only 9 mers or nanomers were associated within this groove. So, in the class 1 molecule, there is only space for a 9 amino acid peptide, as shown here. peptide residue 1, 2, 3, 4, 5, 6, 7, 8 and 9, beyond which it could not be accommodated in this space. So, there were only 9 amino acid peptides sitting in the class 1 groove, as opposed to the class 2 molecule, which had open ends at these two places, and therefore, as I told you, I drew the line of the peptide longer than the groove, and they confirming this, they actually found that the peptide that was sitting within this groove extended beyond 9 amino acids up to 13 to even larger. More than 13 amino acids could be accommodated in the class 2 groove, basically because the class 2 groove was open. So, looking at the properties that was associated with the acid eluted peptides from class 1 molecule, they found that especially this is just an example for one class 1 molecule called as the H2K of D. Now, K is also a class 1 antigen and the D is the haplotype. This just designates the primary amino acid sequence of this H2K of D molecule. They found that in the groove the peptide had tyrosine always in the second position, always tyrosine, and in the 9th position there was always isoleucine or leucine. So, in other words, all peptides that were eluted from the K of D molecule had tyrosine in the second position and isoleucine or leucine in the 9th position. So, one could actually predict by looking at the protein amino acid sequence, or for that matter the nucleotide sequence of the H2 K of D gene, as well as the nucleotide sequence of an antigen, of a putative antigen or a putative protein. If it had tyrosine in the second position spaced later at the 9th position by isoleucine and leucine, one could predict that it was a putative motif for the H2K of D MHC molecule. When they looked at the amino acid residues that were present between tyrosine and isoleucine and leucine, they found these residues given in blue being present in more molecules that were isolated. That is, majority of the acid eluted peptides had these residues and therefore, a strong presence, as opposed to weak presence of some of these residues in many of the molecules that were eluted from the H2K of D molecule. So, this is called as the allele specific motif, because this motif having Y tyrosine or isoleucine or leucine as the dominant anchors was present only in the H2KD molecule. If one was to look at H2DD, or if one was to look at H2KK, or if one was to look at a different haplotype, the presence of allele specific motifs was indeed demonstrated in all of them. However, it was demonstrated that the residues would be different, and therefore, the term allele specific, that means they would be specific to a particular allele of the class 1 molecule. The same sort of principles having the pockets and the binding to certain areas in the floor of the MHC molecule was also present when looking at the class 2 molecule. So, there are certain broad rules that are obeyed when this particular peptide is bound to the MHC groove, whether it is class 1 or class 2. Now, despite the prediction of the presence of this allele specific motifs in particular antigens, it does not however mean that all those predicted motifs would find their way into the peptide binding cleft, basically because the antigen presenting cell would vary in their capacities in order to produce those motifs or clip them at the exact positions that was required for them to have tyrosine in that position and isoleucine or leucine in the case of H2KD molecule. And therefore, the presence of these particular amino acid residues and the presence of a particular motif does not actually mean that it is finally finding its way into the peptide binding group. Another implication of all of the facts that we have learned in the lesson today is that these peptides would vary in their affinities of binding to the class 1 or class 2 molecule. The stronger the affinity, the stronger is the activation by the T cell receptor. The weaker the affinity, the less is the activation by the T cell receptor. So, if one had a peptide that would bind with a very strong affinity, it could actually trigger a self immune response, because the MHC is blind to the self derived antigens. In other words, the peptides that are binding to the class 1 or the class 2 group can be derived from non-self antigens or foreign antigens or pathogens that are infecting, as well as those that are self proteins that are there within that particular cell. In fact, both self derived peptides and non self derived peptides have actually been eluted or have been isolated from the MHC molecules, both class 1 as well as class 2. So, in other words, this MHC molecule is actually blind to the fact that it is self or non self. It is just that the self molecule is binding to a peptide that is generated within the antigen presenting cell. The specificity of self and non self is actually conferred by the T cell receptor, because T cells learn to distinguish self from non self when they are differentiating within the thymus. Although this learning process is not perfect, in that T cells bearing T cell receptors that recognize self and self peptides also escape into the periphery. However, they are less frequent in number. So, therefore, going on further, just to look at some properties of the MHC1 and MHC2 which is described in this slide. One of the important thing being that the MHC1 is ubiquitously found on most cells except those of derived from the central nervous system or the neuronal cells, which express very little MHC on the surface, because these neuronal molecules need to be protected from the immune response. One should note that the presence of MHC on the CNS cells, although less, could be brought about by a variety of regulatory factors. The presence of the 6 binding pockets, the presence of the allele specific motifs, and the number of residues actually binding or contacting the peptide, and that which contacts the T cell receptor and those that contact both peptide antigen as well as the T cell receptor is given in this slide. Excuse me. Going on further to the next slide, you find that MHC2 is present only on the antigen presenting cells or those cells that present the antigen, like professional antigen presenting cells, dendritic cells, and so on and so forth. These have the ability to bind antigenic peptides that are longer than 9 amino acids, and that the helical region of class 2 alpha 1 domain has an extended chain at its amino terminus and allowing it to be open, and therefore, allowing it to bind peptides of a longer nature. So much for the properties of the MHC1 and MHC2 molecule, which can other more details can be found in established textbooks like Kubi and so on and so forth. Going on further, to look at the applications of the MHC molecule and how it can be applied to the skin graft or the graft rejection or the grafting process. Now, the grafting process is not simple. The donor and the recipient have to be HLA typed in order to match for the HLA. If the HLA is different between the donor and the recipient, it would lead to an immune response and therefore, a rejection reaction. So, in order to look at what type of HLA molecule is associated during the grafting process, one has to type for the HLA, and this is called as HLA typing. How was this facilitated in those days when the modern recombinant DNA methods like RFLP was not available? This is called as the microcytotoxicity test. What was observed is that women who were multiparous meaning multiple pregnancies, they had the presence of antibodies to the father of the fetus, to paternal antigens. The fetus derives half its components from the father and half from the mother. So, you have both the paternal MHC molecules and the maternal MHC molecules being inherited and codominantly expressed on the fetus. This leads to the recognition of the paternal MHC molecules expressed on the surface of fetal cells by the mother, which makes antibodies. The mother makes antibodies to these paternal antigens, and these were discovered and characterized as to the specificities that they bound, and these were catalogued in a serum bank, and this was actually what could be used in order to type a donor and a recipient for the HLA type. In addition to that, they observed that patients who had been transfused multiple times with blood also had antibodies to the lymphocytes or the minor fraction of the lymphocytes that were taken in during the transfusion, which got transferred in the transfused blood. So, these were also used to type HLA molecules or for HLA typing. So, how did they use these antibodies for the HLA test? So, if you look at this slide, one isolates the lymphocytes or the mononucleocytes from the buffy coat of the blood that was drawn from a recipient or from a donor, and centrifuging it on what is called as phycol. This phycol material actually enables the isolation of the lymphocytes, the mononuclear cells, as a buffy coat, which comes at the junction of the phycol and the aqueous layer of the blood. This is basically a density dependent sedimentation. This buffy coat that is isolated is then put into plates, is called as Terasaki plates, and reacted with the typing sera that would be available in serum banks, which had already been characterized. For example, in this case the lymphocytes that were reacted with a B3 positive cell with an anti-HLA B3 which had already been characterized, as opposed to an anti-HLA B7 anti-sera that had been characterized to bind to B7 molecules. This antibody bound to the surface antigen HLA molecule that are expressed on the surface, and after this incubation one adds complement, and because complement binds to this antigen antibody complex on the surface, it would punch a hole into this cell as we saw in the previous lectures, and when you put a dye like trypan blue, it would stain the cell blue, and therefore, if you had a positive microcytotoxicity test, these cells would be positive for HLA B3. So, when you use a panel of anti-sera, one could actually type the person in terms of what type of HLA molecule is present on their lymphocytes, and then match for the donor and the recipient. So, this is a microcytotoxicity test that was used for typing individuals in those days before the availability of modern methods. The modern methods of course look at the DNA sequence, and because the HLA complex or the MHC complex is allelic in nature, you have variations in the HLA nucleotide gene nucleotide sequence. So, one could synthesize probes and anneal them to those regions which would vary between individual to individual, and once the sequence, if you know the sequence of the probe, you know the sequence of the DNA that it binds to, and therefore, you can type the individual by using these different recombinant DNA probes. Now, looking at the DNA of the MHC loci in different animals, we found the red stands for the class 1 molecule and the blue stands for the class 2 molecule. You find certain differences in the way they are organized in different animals. For example, the mouse, as we saw earlier, had the class 2 between the class 1 loci, the K and D forming the class 1 locus, and the I region standing for or coding for the class 2 antigen. As opposed to the mouse, if you look at the HLA antigen, you find the HLA antigen being characterized by the A locus, B locus and the C locus. These A, B and C are class 1 molecules, and the D locus stands for the class 2 MHC class 2 molecules. So, these are actually written like this: HLA-A and the allele that follows for the A molecule, for instance, like HLA-A3, A7, A4, A4, or for that matter HLA-B7, B3 and so on and so forth. As opposed to the class 1 molecule, the HLA is written as D. There are subloci within the D. So, they are DP, DQ and DR. So, you have HLA DR3, DR4 or DP4 or DP3 with a number and so on. In certain cases, you will find that the HLA DP, DQ and DR is usually is in rare cases represented with a small w, HLA DR W something. This W stands actually for workshop. This workshop meaning that this particular allele is not yet established, and when it is established by different immunologists all over the world, they drop the W and it becomes a definite specificity like DR3, DR4 and so on. So, looking at the DNA or the genetic map of the human and mouse MHC complex, you have the human HLA being found on the short arm of chromosome 6 and mouse H2 found on chromosome 17. So, this is the details of the MHC region that is found in different textbooks. So, the same thing, if it is not legible in this slide, one could refer to Kobe and you would find the same information. I would like you to note the following point, which is circled over here, TNF A and TNF B. Now, TNF stands for tumor necrosis factor, which is a cytokine that is found on the same region on the same chromosome as that of the MHC. And of course, you have the MHC3 region as we learnt earlier, having the hydroxylase and the complement proteins and other proteins that are found on the MHC genetic map. The finding of the genes coding for certain cytokines like TNF actually points out to the fact that these cytokines could be coordinately regulated along with the MHC molecule. In other words, those regulators that pushed up the MHC molecule or induced the MHC gene would also have the ability to push up the TNF under similar circumstances. So, these molecules would be coordinately regulated. Now, TAP is also found in the same location, transporter associated with peptide antigen, which is involved in antigen presentation and processing. So, LMP is another molecule that is involved in the processing. In addition to the HLA A, B and C, which is shown in red, you have the class 2 antigens shown here like the DQ, DR and DP over here. This is shown for the mouse here, and you find these are the K region, the D region and the L region, and these are some of the minor histocompatibility antigens that are associated with the innate immune response. The TNF also being found similarly just on in the same location, just like the human molecules. The offshoot of all these observations with these HLA typing, they found that certain HLA alleles was always associated with certain diseases. So, these are called as HLA disease associations. In other words, certain HLA alleles bearing humans would always be associated with certain HLA diseases. When they had done all this typing for several human beings, like several patients who come to the hospital, they found that these HLA disease associations could be defined by two terms called as the relative risk and the absolute risk. The relative risk is the chance an individual having the disease associated HLA antigen has of getting the disease compared to one who lacks that antigen. So, this is actually calculated by the patients who are possessing that particular HLA allele, divide that by the patients who are not having the HLA allele, you divide this entire thing by the controls who have that particular HLA allele, which is again divided by the controls who lack that particular antigen. So, this is called as the relative risk. So, relative risk is an important parameter in order to advise patients whether they were at a risk for getting a certain kind of disease. In contrast, the term absolute risk actually gives the chance an individual has of actually getting the disease who has that particular HLA allele. So, AR is defined as the patients who have that antigen divided by the controls who have that antigen multiplied by the prevalence of the disease in the population. For example, if you have a vector that is contributing to the spread of the disease like mosquitoes, if you have mosquitoes, more of those mosquitoes in a particular location, your absolute risk would be higher. So, therefore, looking at these HLA disease associations, you find that certain HLA alleles are always associated with a particular disease. For example, congenital heart malformation is consistently associated with a higher relative risk with HLA-A2, and coming down to ankylosing spondylosis, you see the relative risk being 90.1 for HLA-B27. In other words, those patients or those individuals having HLA-B27 as their allele have a higher risk for suffering from ankylosing spondylosis. Similarly, insulin dependent diabetes mellitus, you find that people who are having DR4 and DR3 allele or the primary amino acid sequence corresponding to the class 2 molecule DR4 and DR3 would be more risky of getting this particular insulin dependent diabetes. Now, as a derivative of these observations, you find that there is a linkage disequilibrium. What does this mean? This means that during random matings, the frequency of a given allele at one locus of the HLA with the second locus of the HLA should be the product of the two frequencies according to statistical principles. However, we find that it is not so in many situations. For example, HLA-B3 which has a frequency of 0.9 in the population and HLA-DR3 which is listed as having a frequency of 0.12 should have an overall frequency of 0.01. That means, HLA-B3 and HLA-DR3 coming in the same individual, but it is much higher. Instead of O1, it is much higher being O7, which means that these two alleles are for some reason being derived in an associated manner. What is the reason for this association? It is due to selective advantage. Perhaps these two are conferring a selective advantage for the immune responses against certain kinds of pathogens or certain kinds of diseases. It also indicates certain migration during the history of man and admixture of these populations. In addition, certain populations are noted for their inbreeding or marrying within the families, like close cousin marriages that happen in India. These are the reasons why the certain HLA alleles are associated, other examples being HLA-A2 and B12. So, what we have seen now in these lectures is the nature of the MHC molecule and how it is involved in the recognition of the peptide antigen, and how the peptide binds with varying affinities to different alleles of the MHC molecules. And the concept that needs to be learnt over here is that due to this variation in affinities of peptide binding to the different alleles of the MHC, and taking in or remembering that we all have different alleles, it is possible that some of these alleles that we inherit actually bind to certain antigenic fragments better than the different kinds of other kinds of pathogens, leading to a certain kind of inequality in how we are susceptible or resistant to particular kinds of pathogens or viruses. For example, if I came down with a particular virus, or children sitting in a classroom can come down with cough, but a different child sitting in the on the same bench in the classroom perhaps may not come down with the same cold and cough as his neighbor. This perhaps is one of the reasons as resulting to this kind of distribution or non recognition, because HLA allele that a person who is coming down with the cough may not be binding to the peptide as efficiently of that particular pathogen that is causing that cough, as opposed to a person where there is a good affinity of binding to the fragments that were derived from that infection, and therefore, there would be a better immune response in a resistant individual than a susceptible individual. But this is not across the board. So, some of us are resistant to certain pathogens, but are more susceptible to certain other pathogens, and therefore, the diversity in pathogen recognition and due to the diversity in HLA in the HLA alleles. This then leads us on to how these HLA alleles are going to be recognized by the T cell receptor, which will come in the next series of lectures. Thank you very much.